In the right condition, even the smallest life forms can grow to change the world. Welcome to AHS Guide, your one-stop destination for clear, concise and exam-focused lectures on the 5 subjects of a first-year RGUHS syllabus. Here, we simplify the complex topics into easier explanations, and the notes and revision tips shall also be provided to study smart and not harder. We'll explore how microorganisms multiply, the factors that influence their growth, and the essential nutrients they need to survive and thrive. From understanding the bacterial growth curves to learning about the nutrient requirements and the culture media, this topic forms the foundation for the microbiology and is essential for both clinical practice and research. By the end of this lecture, you will be able to understand how the microorganisms grow, but also how they are controlling their growth, which is critical for medicine, industry and public health. Without further ado, let's move into the contents of the growth and nutrition topic of microbiology. Growth and nutrition is the first topic. Secondly, we will be covering about the multiplication of bacteria. Third topic is culture media and culture methods. So in growth and nutrition, we'll be studying about what are the conditions required and the factors that affect the growth of bacteria. In nutrition topic, we'll be learning about what are the requirements for the bacteria to multiply, the nutritional requirements the bacteria consumes from a culture media. Now, the multiplication of bacteria tells about how the bacteria reproduces or multiplies in a culture media or a laboratory environment. In the culture media and culture methods, we'll be learning about the contents that are involved in the growth medium or the ones which are allowing the bacteria to multiply. The introduction part of my bacterial nutrition and growth. So the minimum nutritional requirements for a bacterial growth requires the carbon, nitrogen, hydrogen, oxygen, which act as the 4 basic elements for the growth of bacteria. Some inorganic salts might also be involved in the bacterial growth. Now, the classification of bacteria based on nutrition, we have autotrophs, heterotrophs. Autotrophs is also called as lithotrophs because they are on the earth. These autotrophs can synthesize whatever essential metabolites they require from the atmospheric carbon dioxide and nitrogen. So they derive nutrients from the atmospheric carbon dioxide, the CO2 present in the air and the nitrogen that is present in the air, and produce or synthesize or cook their own food. Now these obtain energy by the process of photosynthesis. Some get energy by photosynthesis, the others might go through chemolithotrophs or the chemoautotrophs. Now by photosynthesis, the photolithotrophs, they consume light from the sun and synthesize their own food along with carbon dioxide and nitrogen like in plants, and some also oxidize the chemical compounds and break down the chemical compounds for their nutrition. So these are called chemoautotrophs. Moving to the second type of nutrition-based classification of bacteria. Heterotrophs are they don't synthesize their own metabolites. They don't cook their own food. So they are dependent on certain organic compounds, and sometimes these heterotrophs can be pathogenic as well or parasitic. So they derive nutrition by killing the other bacteria or an organism. Now these are evolved with animals where nutrients are readily available. So they are very strongly related with the animals because their animal nutrients are readily available for the bacteria to grow easily. Now the bacterial growth is dependent on the bacterial cell division. The cell division happens by binary fission. Binary fission, as the word says, bi meaning 2, and fission is breaking down or dividing. So one cell is divided into 2 is the name that is suggested for a binary fission. Now the steps of a binary fission involve the DNA replication happens. You can see the blue color and the green color region, that is the nuclei of the cell which are dividing. Now here, for the first step is the DNA replicates, that is there is multiplication of the DNA happening, which is identical to one another, and they form a circular double-stranded DNA molecules. How does the DNA replicate? It replicates or forms a mirror image of its own, which is two similar circular double-stranded DNA molecules. Now the transverse septum formation from the cell membrane happens, that is the cell membrane starts constricting at the ends. Here you can see in the image the cell membrane is narrowing or constricting at the mid of the two nuclei that has been formed inside the cell. Now these deposition of cells separates the cell into two daughter cells. Some bacteria might remain partially attached, like in diplococci, the pneumococcus. So the two cells are attached together, or in the form of chains as in streptococcus. Either the bacteria can completely divide into two individual cells, they can be attached as in diplococci, or they can form chains as in streptococci. So these are the few examples of binary fission that is happening inside the different types of bacteria. Now the clinical significance of this bacterial or binary fission is the morphology helps the bacterial identification. So now when you see a certain type of cell, you're going to able to identify the cell that this is the type of the cell is because of the binary fission. So certain bacteria have a same kind of nuclear material, same type of cell membrane that is observed because of the identicality or the mirror image that is being formed because of the binary fission. Now the generation time is usually for one bacterium to divide into two daughter cells under optimum conditions, that is a proper nutrition is available, a proper environment for the DNA replication to happen, everything is at optimum, at a correct conditions, then only the bacteria is able to generate its daughter cell is approximately around 20 minutes. So medically important bacteria take approximately 20 minutes. Like in case of Mycobacterium tuberculosis, it is approximately 20 hours, and in Mycobacterium leprae, it is approximately 20 days. So different bacteria have different generation time, or the time that they require to multiply. Now most of the bacteria take approximately 20 minutes, but there are certain cases wherein the bacteria takes longer hours to divide into two daughter cells, as in tuberculosis causing bacteria, which TB causing bacteria, which is Mycobacterium tuberculosis. It takes approximately 20 hours. So it requires one day to generate into two daughter cells. In case of Mycobacterium leprae, which causes leprosy, it takes approximately 20 days to divide into two daughter cells. So different bacteria have different generation time. Now the implication of this generation time is that there are fast growers and there are slow growers. So the fast growers, which is approximately 20 minutes, they form colonies in just 18 to 24 hours. In just one day they can form colonies. But in Mycobacterium tuberculosis, they take weeks to grow as a colonies, and Mycobacterium leprae can take months to grow as a colony. So colonies are I'll show you an image of the colonies. Colonies are a group of bacterial growth on a culture medium. Now we have learned about the two aspects of the growth of a bacteria, which is one is bacterial cell division, the second one was the generation time that determines the growth of the bacteria, and the third one is the bacterial count, so the number of bacteria that is present in the culture medium. So total count is the total number of bacteria, whether it is living or dead, are taken into consideration. So it is measured in the microscope by counting the chambers of the bacteria. Now to viable count is the count of living bacteria alone, and this method is viable count is done by dilution method or plating method. So in dilution method, the liquid media containing bacteria is diluted, and the count of the tubes with the growth is determined. Now here the inoculation of dilution in the liquid media, meaning the liquid mediums which have the bacteria inside it is diluted, and the number of times the bacteria has reduced its count as you dilute the culture media, the count, the very last tube wherein the bacterial growth is stopped is the amount of the living bacteria that is present in the number of tubes here. So here you can see the liquid media. So here they are going to see how till what dilution the bacterial growth is possible. Those many tubes are counted and the growth is determined. Now in plating method, the diluted samples on solid media colonies are counted. So here is the culture plate wherein the bacterial smear is made on the plate and the bacterial growth is observed. The colonies are counted, whatever bacterial colonies are there are counted, and the living bacteria is determined. Now what are the uses of counting this bacteria? It helps in water testing. So in case of the water is infected or the water is said to be not pure or not pure of any microorganism is presumed by testing the water by smearing the water, taking a smear of the water and doing a culture plate testing. So after counting the number of colonies, you can determine whether the water is clear or is being infected by any bacterium. Now another use of doing the bacterial count is to diagnose the UTI. The urinary tract infection can also be determined by taking a sample of urine and smearing it on the culture plate, and the colonies are observed for any urine infection caused inside the urinary tract can be determined. Now the bacterial growth curve is another factor that is helping to determine the bacterial growth. Now when the liquid medium is inoculated, meaning the liquid medium with the culture medium wherein the bacterium grows is determined on the 4 different phases. Now the first phase is lag phase, second phase is log or exponential phase, third phase is stationary phase, and fourth phase is the decline phase. Here the cell while growing, the bacterial cell while growing, it goes with the small colonies. It suddenly increases the number of colonies and then it goes a constant phase. After the constant phase, it starts at a certain stage it starts declining the growth. Now you can see here is the bacterial growth curve. This is an important topic and the question might arise from bacterial growth curve. So it might be around 5 marks question in microbiology. It is very much important to remember what are the different phases of bacterial growth curve. So under lag phase, here the bacteria is trying to adapt to the liquid medium and enzymes are being synthesized in order to increase the number of bacteria inside the liquid medium. So whatever bacteria that is present inside the liquid medium, whatever bacteria is inoculated in the liquid medium, it starts to adapt to the liquid medium and the enzymes are synthesized by the bacteria, thereby increasing the number. In log phase or exponential phase, the rapid cell division happens and there is geometric progression, meaning the cells have adapted nicely to the liquid medium and they have started to rapidly or fastly undergo cell division or binary fission or any type of cell division methods and have increased to maximum potential by increasing the number of bacteria inside the liquid medium. Now the stationary phase is where the nutrients in the liquid media have started to become less for the growing bacterial community. For the bacterial community, the nutrients have been stopped and there is accumulation of toxins. That is, whatever bacteria after taking up the nutrition and is being releasing the toxins are getting accumulated in the liquid media. So this balance between cell death and new cells has happened at this stage. So the stationary phase is where the nutrients have already been used nicely and some certain toxin are being accumulated, thereby causing the balance between both cell death and new cells. This phase is called as stationary phase. Now the last phase is the decline phase where cell death exceeds. So there is more number of cell death than the cell growth because the toxic products and autolysis, that is the cell death that is happening inside the liquid medium, is increased and there is more number of bacterial cells that is being dead. So these are the 4 different phases of how a bacterial growth is represented in the form of a curve over here. So this is very important. Understand the concept. You can rewind back to where I started the bacterial growth curve, understand the different topics and the important words that are being used inside different phases, and start writing the answers by your own without seeing. Thereby, it will help you how to write the bacterial growth curve as an answer in your exams. Now moving to the next one, which is morphological changes during growth curve. So what are the changes that you observe when the bacteria is growing on contrast with the bacterial growth curve? So here is a theoretical representation of how a bacteria grows. So the bacterial growth curve is put in textbook, but when you're doing it practically in a lab, you will observe certain changes of the bacterial growth inside the medium. Now here at the end of the lag phase, the maximum cell size is seen. So lag phase is the first phase wherein the bacteria is starting to adapt to the environment and there is increased cell size. The cell size becomes bigger. And at the log phase, there are small uniform staining. So the whatever cells that are being at maximum size are starting to become small and are at the same stain that are being absorbed by the bacteria, followed by stationary phase where both new cell and the cell dying both are at the same balance. There the cells are appearing to be gram variable because the dead cell does not absorb much of the stain and the new cell absorbs more of the stain. And there is irregular staining because of this. The sporulation also happens over here. Sporulation is the spore formation, that is because of the unfavorable condition or nutrition that is not left inside the medium. The sporulation is happening. Sporulation is a type of reproduction that is seen in bacteria. When an unfavorable condition happens, the bacteria starts to form a thick covering surrounding it and starts dividing inside the thick covering. So that is a sporulation. And exotoxin production. After the stationary phase, the cell death is seen. Because of the cell death, there is more of toxins that are produced inside the culture medium. After the stationary phase and the cells start dying out, the decline phase is seen and the involution forms common. So there is no further evolving of the bacteria because of the death phase of all the cells. So you can see the image of how the different phases can be seen. This can be the lag phase, the log phase wherein maximum size of the cell is attained. Then the cells keep on multiplying till the end wherein the cell death occurs. So that is the morphological changes during the growth curve. Now the next feature of bacterial growth is the batch culture versus the continuous culture. In batch culture, the growth stops after the nutrition is depleted. In continuous culture, the nutrition is being provided continuously without any replenishment, and the cells that are dead are going to be removed as the nutrients are taken out, and there is sustained growth. So there is continuous growth. As the word says, batch culture is where the nutrients is stopped and is at smaller quantities. In continuous culture, there is nutrition that is being given again and again after the cells have attained a lag phase and is at a stationary phase. The nutrition are again provided in order to keep the lag phase and log phase running. So that is continuous culture. Now the devices that are used for batch culture and continuous culture are chemostat and turbidostats, and they are used in industries and research for keeping the batch culture and continuous culture in vivo is growing inside a bacterial body itself. In vitro is growing in the laboratory. Now in host tissue, the bacteria cannot form a batch or a continuous culture. So nutrients are abundant, though the nutrients that are provided by the host organism to the bacteria is more. The host might defend against the bacterial growth sometimes also. when the host defends the mechanism of the growth, the bacteria can be dead inside the host cell because of the immune system of the host cell. Now, what is a host tissue? Host tissue is where the bacteria is deriving its nutrition from. Now, the host tissue acts as a nutrient medium and provides the nutrition abundantly, continuously. Now that we have seen the bacterial growth, we'll be seeing the next topic, which is bacterial nutrition. The first point under bacterial nutrition is the principal component. The cell contains approximately 80% of water inside it. So the cell weight of a bacteria is 80% of water, and the rest 20% is the nutrients, the bacterial nucleus, and the cell organelles present inside it. Now, the minimum nutritional requirement for a bacteria should have certain components, which are water, sources of carbon, which is like carbohydrates, the glucose, any of the components of carbon, and the source of nitrogen, inorganic salts, sulfur, phosphorus, and the trace elements such as sodium, potassium, magnesium, iron, or manganese. Now, the growth factors that help that work as a nutrition to the bacteria are the bacterial vitamins, such as the vitamin B group, which involves thiamine, nicotinic acid, riboflavin, pyridoxine, folic acid, and vitamin B12. We'll be studying about the vitamin B group in the biochemistry chapter. Till then, just know that the vitamin B group is essential for the bacteria to obtain nutrition. So these bacterial vitamins or growth factors can be essential or accessory. Essential is it is impossible for the bacteria to grow without certain bacteria growth factors. Those are called as essential growth factors. The others are accessory growth factors, which might enhance the growth but are not so essential or is not required essentially, or in the absence of these growth factors also, it can be let the bacteria to grow efficiently. Now, the significance of these growth factors is that they provide a knowledge about which all nutrition is required by the bacteria to grow in a laboratory. So you'll be able to prepare a culture media based upon the specificity of nutrition requirement of a bacteria. The environmental factors that affect the bacterial growth. So the various factors that cause the deteriorant or increase the growth of bacteria can be determined by various factors. So the first factor is moisture and desiccation. As we have learned that the principal component of the bacterial cell is approximately consisting of water, which is 80%. Approximately 80% of a cell contains water, and in absence of water, the bacterial cell growth might be detrimental or it can die. So the drying effects depend upon various type of body cells. In bacteria such as Treponema pallidum and Neisseria gonorrhea, which are the two bacteria which cause diseases in the human body, like the Trypanosoma causes the sleeping sickness and Neisseria gonorrhea causes the sexually transmitted diseases, they die quickly if the water is not present or the moisture is not present. In case of Mycobacterium tuberculosis and Staphylococcus aureus, they can survive for weeks, but later, if the water is not present, they'll eventually die. So these are the drying effects of if the water is absent. Now, how do you preserve a microorganism? Whereas in case of bacteria, microorganisms such as the polioviruses or any other organism that is used as antibiotics or vaccines, they are freeze dried and kept. So freeze drying keeps the microorganisms from growing. So it is intentionally made to stop the growth of the microorganism for the beneficial effects that they provide to the human. So that is how preservation is useful. So preservation keeps the microorganism in certain phase where they don't multiply in excess amount or cause any diseases. Now, the clinical note that you have to remember is that Neisseria needs a transport medium, which we'll be learning in the upcoming sessions. For the Neisseria to be transported, when a Neisseria bacteria is taken as an smear from the human body, it is transported to laboratory with certain culture media, which is called transport media, in order to keep it moisturized. Otherwise, the bacteria will die quickly if the moisture is taken out, and hence there is a need of transport media for Neisseria gonorrhea. But for Staphylococcus aureus, there is no requirement of transport media because they can survive for weeks without moisture. Now, the oxygen requirement can also be a boon or bane for the bacterial growth. In case of aerobes, they constantly require oxygen. Some aerobes can be obligate or facultative or microaerophils. These obligate aerobes can grow only in the presence of oxygen, and if there is no oxygen, they eventually die. Some of the examples of obligate aerobes are Pseudomonas aeruginosa, and the facultative anaerobes which can grow with or without oxygen. If the oxygen is present or absent, it doesn't matter for the facultative anaerobes, and they grow with or without of them. So most of the pathogens are said to be facultative anaerobes. Microaerophils are the ones which require trace amounts of oxygen. It is not that higher amounts of oxygen is required. Small amounts of oxygen can also help them survive, such as Campylobacter and Helicobacter pylori. Coming to the second type of oxygen requiring bacteria, which is oxygen based upon oxygen requirement, the anaerobes, which grow only in the absence of oxygen. So there should be no oxygen for their growth. So Clostridium tetani is one such example which grows without oxygen. This oxygen is toxic to these obligate anaerobes because they can grow only in absence of oxygen. The carbon dioxide is another factor which determines the growth of bacteria, wherein it is needed in small amounts because it is a nutrient medium, and it is normally available in the air or the atmosphere or by metabolism, that is during the digestion the bacteria synthesizes carbon dioxide by itself. Some bacteria like Brucella abortus needs 5 to 10% of carbon dioxide, which is referred to as capnophilic bacteria, because they take in carbon dioxide for the survival. The fourth point is based upon temperature, the factor which determines the growth of bacteria. Here the optimum temperature required may vary for different species. That is, different species might require different temperature for them to grow and survive. So most of the pathogens or most of the microorganisms require 37 degrees Celsius, which is optimal for the growth of certain disease-causing bacteria or microorganisms in general. So based upon the temperature requirement, the bacteria can be divided into mesophils, psychrophils, and the thermophils. So mesophils, the microorganisms grow in between the range of 25 to 40 degrees Celsius, and most of them are pathogens. Psychrophils are less than 20 degrees Celsius, that is, the temperature is lower than 20 degrees Celsius, and usually the bacteria grow as soil or water saprophytes. The saprophytes are the ones that consume from dead and decaying organisms. The thermophils are the ones with high temperatures, that is 55 to 80 degrees Celsius of temperature is required for the growth of the bacteria. One such example is a Geobacillus stereothermophilus. So these require higher temperatures. Geobacillus stereothermophilus might be growing near the hot springs. Now the clinical note that you have to remember is that the pathogens die at 55 to 65 degrees Celsius, and the spores require 100 to 120 degrees Celsius. Spores, because spores are formed during unfavorable condition, and the unfavorable condition is produced when the outside climate is not optimum for the bacterial growth. Spores are unfavorable condition reproductive function that is seen in the microorganisms. Now to sterilize or to completely remove the pathogens from any certain organism, sterilization is performed. So the sterilization is done by producing a temperature of 55 to 65 degrees Celsius to kill the pathogens, and for the spores to be killed 100 to 120 degrees Celsius. Now preservation is also essential in order to keep the organisms of future reference or research. The preservation is done. So preservation is done with lower temperature, about 3 to 5 degrees Celsius, or minus 30 degrees Celsius to minus 70 degrees Celsius. So the clinical note involves sterilization and preservation. Now pH is another factor determining the growth of bacteria, and most of the pathogens grows at the alkaline nature, which is 7.2 to 7.6 pH. So some of the exceptions which grow in acidic pH, some in alkaline. Most of them grow in neutral pH, but few of them grow in acidic and alkaline pH. One such is lactobacillus, which grows in acidic pH, and Vibrio cholera, which grows in alkaline pH. The strong acids or alkalis might kill the bacteria, except for Mycobacterium, which can survive in both strong acids and alkalis. So the clinical note that we have to note here is that the Vibrio cholera can be grown in a basic medium, and the Terov method is used to diagnose the tuberculosis in acidic or alkaline pH. So because Mycobacterium can still survive in the acidic and alkaline medium. If it is any other bacteria that is present, it will be easily killed because of the acidity or the basicity of the culture medium. Now here light is also a factor that determines the growth of the bacteria. Most of the bacteria grows in the darkness. They are sensitive to ultraviolet rays and the radiations that are emitted by the sun, and hence the bacteria is assumed to be growing in the darkness. Some bacteria like Mycobacteria produces pigments when exposed to light. So they are photochromogenic, that is they absorb light, and the light is emitted after exposure to them because of the presence of a pigment which is photochromogenic. Now the osmotic effect is another form of factor wherein the water moves from higher concentration to the lower concentration. Bacteria can tolerate a wide osmotic changes, that is it can shrink or expand, swell up into at higher concentrations of osmotic changes, but because of the presence of the strong cell wall. If the bacteria is put in a hypotonic solution wherein the more salt solution is present, the water inside the cells flows out and they shrink up. So this process is called as plasmolysis. When a bacterial cell is put in a hypotonic solution where the salt is less, the water moves into the cells and causes the swelling or rupture of it, which is also called as plasmoptosis. The gram-negative bacteria is affected more by the osmotic effect than the gram-positive bacteria, because gram-positive bacteria have an extra myelin sheath around them protecting against the effects of osmosis. And hence, gram-negative bacteria are mostly affected by the osmotic effect. Now, the mechanical and zoonotic stresses. Here, the bacteria can be disrupted by vigorous shaking with the glass beads. So, bacteria and the glass beads are put into a test tube and nicely shaken. So when the shaking happens, the bacteria can be disrupted and can die. Sometimes ultrasonic vibrations also can cause the disruption of the bacteria. The next topic, which is bacterial metabolism, is the bacterial digestion, elimination and absorption of nutrition. So we'll see the definition of what metabolism is. So the series of changes of substances of carbohydrates, proteins and fats within the bacterial cell from absorption to elimination. So whatever changes that happens from absorption to the elimination of whatever nutrition that is consumed as in carbohydrate, proteins and fats that is taken up by the bacterial cell, it starts absorbing, breaking it down and eliminating the waste products. That process is called as metabolism. And the energy generation after metabolism is different for different organisms. For aerobes, it uses oxygen and accepts the hydrogen that is produced during the metabolism. For anaerobes, it uses hydrogen acceptors which are not oxygen. So it can be sulfur, it can be nitrogen, it can be any other compound that is accepting the hydrogen. Now, oxidation or aerobic respiration. In here, the ultimate hydrogen acceptor is oxygen. So, in aerobic organisms, due to the presence of oxygen around it, the water molecule or the H2 hydrogen that is produced during the process of metabolism is accepted by the oxygen. So, the end products that is obtained here are carbon dioxide, which is CO2, and H2O, which is water. So the energy that is produced is by conversion of ADP to ATP by oxidative phosphorylation. So oxidative phosphorylation happens only by the accepting of oxygen. So when the accepting of electrons takes place, the ADP is converted to ATP, producing the energy to the cell. Now the byproducts that are obtained during the process of oxidation are hydrogen peroxide, which can be removed by the enzymes like catalase and peroxidases. So hydrogen peroxide is usually toxic and it is removed by the certain enzymes that is present in the bacteria like catalase and peroxidases. Moving to the second one, which is fermentation. The anaerobic respiration. Here the hydrogen acceptors are nitrates, sulfates or organic compounds. Because there is no oxygen or no oxygen is required for the anaerobic respiration of microorganisms, the hydrogen acceptors has a supplements like nitrates, sulfates or organic compounds that accept these hydrogen that are released during the metabolism of certain nutrients. The process is by series of oxidative and reductive reactions. So both the redox reaction, that is oxidation and reduction, is taking place simultaneously, allowing the accepting of electron and donation of electron easily to be processed out. Now the end products that are obtained during anaerobic respiration are lactic acid, formic acid and pyruvic acid, and the gases that are obtained during the anaerobic respiration are hydrogen and carbon dioxide. The alcohols are also produced during the anaerobic respiration, and that is how fermentation takes place. Energy generation. By substrate level phosphorylation, phosphate introduced into intermediate metabolites and ATP is formed from the ADP. As in case of oxidative respiration, the oxygen acts as the oxidative phosphorylation pathway product, but here the energy generation happens at the substrate level and the phosphate introduced here act as a intermediate metabolites, thereby producing ATP from ADP. Now the organisms that are involved in the fermentation are obligate anaerobes and facultative anaerobes, which require lower amounts of oxygen or no oxygen at all. The toxicity of oxygen to anaerobes, the accumulation of hydrogen peroxide and lack of catalase enzyme and essential enzymes active only in reduced state. So presence of oxygen in anaerobes can cause accumulation of H2O2 or hydrogen peroxide, which can lead to the lack of catalase enzymes, the enzymes which are involved in the breaking down of hydrogen peroxide. These are not present in the anaerobes, and essential enzymes that are active are also reduced into the inactive states. The facultative anaerobes, as in case of the ones which are grown without the presence of oxygen or total absence of oxygen, here the facultative anaerobes can switch to aerobic respiration at times also. The clinical significance in anaerobic respiration is the biochemical reaction. The fermentation process is used to identify the gram-negative bacilli that is present, and the fermentation leads to gives the products acid and gas such as lactic and pyruvic acid, and gases like H2 and CO2 are also important. So the third topic under bacterial metabolism is oxidation and reduction potential, which is also called as redox potential, wherein both oxidation and reduction takes place at the simultaneous process. So the definition of oxidizing agent is the ones which accepts the electron is referred to as oxidizing agent, and the ones which donate the electrons is referred to as reducing agent. So the redox potential
or E is, or the representation as EH, indicates the oxidizing and reducing condition that happens inside the system of a bacteria. It can be measured in millivolts using an electrode, and the amount of oxidation reduction that is taking place due to the accepting and donation of electron. The process generates the certain amount of electricity, which is measured by electrodes and the millivolts. The highest EH or redox potential is oxidized system, and the lowest EH is the reduced system. And the reduced system is considered to be favorable for anaerobes because they accept the electrons and help in the metabolism inside the bacteria. The indicator dyes that are used are methylene blue or resazurin. The colored is in oxidized state and colorless in reduced state. These indicators are used inside the redox potential systems to determine the oxidization and reduction from the naked eye. So when the oxidation takes place, the color is observed, and by the presence of methylene blue or resazurin, the indicators, and it is colorless in the reduced state. Anaerobes are growing well in the low redox potential because of the reducing system that is observed. The clinical significance of redox potential is they are helpful in anaerobic jar production, that is used for anaerobic growth, and anaerobic jar is produced by reducing or accepting all the electrons that are present inside the chamber and causing a vacuum environment, and thereby removing all the oxygen and allowing the growth of the microorganism inside. The redox potential is also used for methylene blue indicator, where blue is aerobic condition and the jar is said to be or the system is said to be colorless in anaerobic conditions. So moving to the culture media and the culture methods, we'll be dealing about them in the next video. Till then, keep watching AHS Guide.